Good afternoon everybody. Now last lecture we have discussing about the intragenic interactions. In this one of the Mendel's deviation we shall study, and this Mendel deviation they are including the two interactions, intergenic interactions and intragenic interactions. These two interactions means first one the intragenic interaction. Now in this interaction this interaction takes place between the same genes or same allelomorphic pairs. With this type of interaction it is known as the intragenic interactions. And now some example we will study. This first example, incomplete dominance, second one is the codominance, and another one is a multiple alleles. With this multiple alleles means when the single gene is controlled the different traits similar as the second one. The intragenic interaction is also similar for this multiple alleles. For in this multiple alleles we shall study this example of blood groups, human blood group or some flies of Drosophila flies. And now in this lecture we are discussing the intragenic interactions, and one important example is the pleiotropy. This first one is the intragenic interaction we will discussing, and now in this lecture we shall study the intergenic interactions. Intergenic interactions. This intergenic interaction it is occurring between the different gene or different chromosomes. For example, now see the structure of chromosomes. Now this is a centromere. This is a one pair of homologous chromosomes and another is this two pairs. And in these two homologous chromosomes, this intergenic interaction it takes place between the different gene or different chromosomes. In this first one, this is a suppose A gene and another in this chromosome having the T or small t, this another gene or alleles, and this interaction now in this gene is interact to another gene and this gene is also interact with in this gene. With this interaction is different. With this one gene they can also interact with other genes. In this type of interaction it is known as the intergenic interaction. In this intergenic interaction it is occurring the alleles of different gene present the same or different chromosomes. Means this interaction is also takes place the same gene or this interaction is also takes place from the another gene. And this intergenic interaction, one of the important example is the pleiotropy. Now this pleiotropy, the pleiotropic gene or pleiotropic interaction is the most important deviation. Now the concept of this pleiotropy means one of the single gene is controlling the different traits. One single gene, this single gene is controlling the different traits. This one, a single gene, they can controlling the different traits. For example, we shall study this blood group, human blood group. This one single trait is a blood, and there are different blood groups like A, B, O, and these are the different traits in this human blood group. Also Drosophila flies, there are different shapes of these wings, and some another example like flower color of pea plants. They can also control one single gene control the different trait, and this term it is known as a pleiotropy. Pleiotropic gene means this one of the important example of this pleiotropic gene in human being sickle cell anemia. This is a disease or one of the disorder. It is takes place in human beings, and this sickle cell anemia it is one of the disease is created, and this one example in pleiotropy, sickle cell anemia. Pleiotropy gene example, sickle cell anemia. This is one of the disease is creating the human beings, and this sickle cell means this sickle cell disease is caused by RBCs. These red blood cells in human beings, the red blood cells found, and this RBC cell, this sickle cell anemia is carrying. This normal sickle cell is a rounded shape structure. This is a normal shape of RBCs. RBCs means red blood cell is a round shape, but sometimes this anemia disease is causing, that time this sickle cell is a sickle like structure or half moon like structure is formed. These are forming this type of shape, and that's why this disease is causes. This anemia disease is causing the human blood groups. At that time, this blood group means this RBC is causing this anemia. That time sickle cell, they are the sickle like structure or half moon like structure, and that's why this causing this anemia disease. And that time the oxygen deficiency takes place, because this cell RBCs is a rounded structure. That's why flow of blood is a regular continuous flow of blood is takes place. But sometimes this anemia is caused, that time this RBC shapes is a curve like or it is a moon like, and that time this blood is not supplied, they are not passes, and that time oxygen deficiency takes place. This oxygen deficiency is occurring by human beings, and this is one of the type of diseases causes of human beings, sickle cell anemia. Now the one of the example, this sickle cell anemia, there are the homozygous or heterozygous trait are formed. The first one of the gene, this HBA, this is one gene, it is a healthy gene or normal gene of, and another is a HBS, disease causing gene, and this gene is also called sickle cell carrier. This gene, now these are the two genes, one is a healthy gene and another is a disease causing gene. In this sickle cell anemia, instead there are some phenotype or these genotypes are forming. The first one, the parents HBA and HBS, it is heterozygous and it is a sickle cell carriers. And another gene, it is a HBA and HBS, it is also the sickle cell carrier. In these two genes, or this normal gene or healthy gene, this HBA is a normal gene and HBS is a disease genes, or it is also anemia disease is forming, the sickle cell anemia. This gene is called HBS gene, and this crossing is takes place between these two genes. That time some genotypes are formed. In this there are some genotypes. This is a parents sickle cell carriers, and they can form the two types of the genotypes. HBA is a one and another is a HBS. In the same condition, HBA is one and another is a HBS. They are forming the means marriages between two the sickle cell carrier. That time this genotypes is a two types of the genotypes, HBA, HBS. Now in the F2 generation is counted. That times F2 with the help of checkerboard method, F2 generation. This one HBA or HBS and another one HBA, HBS, F2, and this crossing is takes place HBA, HBA is a normal gene is formed, means healthy gene is produces, offspring is a healthy. Next one HBA, HBS, this is sickle cell carriers, sickle cell carrier. Next one HBA and HBS is another sickle cell carriers, sickle cell carrier. Now last one HBS and HBS. This gene, it is a recessive gene is forming. These are some are the heterozygous genes and this one is the homozygous genes, and it is a sickle cell anemia disease causes. Sickle cell anemia means this offspring is produced and this offspring is die. Means there are four types of offspring are produces, and that time one is a normal and two is sickle cell carriers and one is a anemics, means causing the anemia disease in the fourth offspring, and that's why the ratio, this ratio is obtaining instead of this ratio is 1:2, 2:1. This ratio is obtaining by the sickle cell anemia disease, causing that time one is a normal, two is a sickle cell carriers and one is a disease. But this one, that time this offspring become die. This is died. This offspring is a because the HBS and HBS, this gene is a homozygous recessive genes are obtaining, and that time this is a diseased gene and they can causes these human beings, that time they become die. This the sickle cell anemia disease is obtaining the human beings, and this is one example solving, and some ratio is obtaining 1:2, 2:1. Now in this chapter we shall also discussing the different points, like first one point we discuss these 7 pair of contrasting characters. Mendel studied this pair of contrasting character, and also the Mendel select these garden pea plants, and some ratios like monohybrid and dihybrid ratio, and some deviations. Mendel studied the some post-Mendelian ratios like intergenic interactions and intragenic interaction,